Rollins Professor at the Rollins School of Public Health, based there in Atlanta, Georgia at Emory University. He is a member of the National Academy of Science Committee on the Applied and Theoretical Aspects of Statistics, and his research involves the development of statistical methods for geographic data, including applications in environmental justice, epidemiology, disease surveillance, spatial cluster detection, conservation biology, and disease ecology. His research has appeared in numerous biostatistical, statistical, environmental health, and ecology journals, as well as he's authored a textbook entitled Applied Spatial Statistics for Public Health Data. And so prior to us getting Dr. Waller underway, I'd like to invite you all to submit any questions that you have for Dr. Waller through the GoToWebinar question submission system, which is located in the little console there off to probably the right of your screen. And you can do that, and then we'll save some time at the end where I'll read those questions off to Dr. Waller. So without further ado, Dr. Waller, the stage is yours.
Great, thank you. It's a pleasure to be here, and I'd like to offer my own wishes for a happy St. Patrick's Day. I hope you're all doing very well in your office basketball pools, because as data scientists, you should be. Today we will be talking about, well, that's what I look like. And we'll be talking about multiple testing, bias, confounding, and missing data, or as a subtitle, what could go horribly wrong with my analysis? I'm going to give a little insight on thinking about team data science and team science in the data science setting, with a little bit of background to distinguish between observational experimental data and prediction versus estimation. To put this in context, I've been watching these webinars over the last several weeks to get a sense of the kinds of topics, and we have a lot of people with a lot of different interests. So I want to set some context. We'll talk about multiple testing, bias, confounding, missing data. I'll try to give an example for each, and sources for additional information, and then wrap it up coming back to team data science. So data science is often presented as requiring skills from computer science, statistics, or an application or subject matter area, which often leads to the battle of the Venn diagrams. This is just one example here in a couple of postings where people have looked at this, but you can see sort of the three scopes. We've got this pinkish color, hacking skills or computer science. We've got math and statistical knowledge, and then we've got the substance area. And in a school of public health, for me, it's a lot of health-related research. Data science is kind of that sweet spot in the middle. Then we could have more than three directions. One of them has a 7-set Venn diagram, which I'd never seen before, so I thought I'd share that with you too. The webinar series, I really appreciate the BD2K program putting this on, because it has provided and continues to provide a forum for introduction across these elements. So if you go to data science seminars within your own discipline, you tend to hear people similar perspectives talking, and I think really where the growth is this crosstalk. So I've really learned a lot from the webinars, and hope you're enjoying them as well. In computer science, there have been webinars on hardware, just even the basic setup of doing big data, also algorithms. And statistics, we've had several on concepts, techniques, Bayesian analysis, and so on. I think it's the intersection of all these where the unique and emerging issues come up. And today, what I'm talking about are trying to draw some statistical concepts from observational studies, often from epidemiology, then how these may scale up, or how they may need to be revised in a big data setting. So we'll talk about definitions, examples, and the scaling issue primarily. I think each and every one of these is wide open for additional work. So I'm not stating that these are problems of a statistical nature and dropping the microphone, but instead we want to have an invitation for the interdisciplinary work and really work those intersections in the Venn diagram. Team data science, there's a lot of interesting discussions going on at conferences and in publications on the intersection between these areas. And one thing that I've heard recently is the discussion of whether a team of data scientists should consist of several people who are identical or several people with complementary skills. I tend to favor the idea of a heterogeneous team because it brings different expertise and experience, also different problem-solving intuition. That's a big deal to me. Are people understanding how do they approach a problem? What do they think the cool part of it is? And if you have different people on the team looking at that from different aspects, I think you get much more creative analytic and interpretation perspectives in your analysis. Interdisciplinary team science can be very tricky if you want to go all the way to transdisciplinary collaborations. I tell my students and colleagues that I think it's a lot like international travel when you work interdisciplinary setting. The language is different, the social norms are different, what the cool journals are is different, how money counts, what you should write into a grant, that all needs to be negotiated. It's also a lot like a middle school dance where you've got multiple groups who are interested and they know they want to interact with each other, but they're not quite sure how to do that yet. And so they tend to cover up the insecurity with a lot of teasing. And we see that in the publications too, of they're sort of trying to establish your own discipline as the key discipline in here. I also recently, I don't have this on the slide, heard a definition that disciplinary work is like an apple. Disciplinary collaborations like a fruit basket, interdisciplinary works like a fruit salad, and transdisciplinary work is like a smoothie. So how do data impact science? That's our question. I mean, our goal in big data to knowledge is to take data, extract information from it, and develop knowledge. So I want to frame this in terms of questions, analytic tools, and answers. And this is something we present in class a lot, geographic information systems class I start with. And I can't take credit for this. John Richardson, who was a toxicologist at the EPA Region 4 office here in Atlanta, once said, it's this simple, and he drew this picture, and I thought it was brilliant and really liked this. So if we start in the upper left, there's some question we want to answer. And I know in a lot of unstructured data mining, there's a lot of talk about hypothesis-free analysis, but I don't think we go in completely blind. I think there are things we're interested in finding out. So they may be hard to articulate, but this first step is articulating what you're looking for, what kinds of things you're looking for. And before running anything or before extracting data or looking for the data, I think it's important to spend some time thinking about what data would you really need to answer that question completely. I do a lot of environmental health analysis, so if I were interested in what's the impact of exposure to drinking water near a hazardous waste site on childhood leukemia rates, I might want to know where every child lives, at what age, how much water do they drink, does their house require this. I could go through a list of all the stuff I'd love to get that I probably wouldn't be able to. But that list is a helpful mental checklist when you take the next step and say, what data are available? We have a wide variety of data types available to us now, so we can get all sorts of things, but we rarely get everything we want, and we often get proxies for things we want. Like I may not know which child is drinking from which water system, but I know where they live and I know where the hazardous waste site is. And I also may be able to find out that that address is listed on the county public water bill. So I know they're getting water to the house, but I don't know exactly what's going inside. So it tells me something, but not everything. So on the lower right-hand side, I get the data I can get and the methods I have available. I do answer some questions as I move up to the upper right. There are questions I can answer with my data and my methods. And probably the most important step here is to go back to the beginning and see how close the question I answered is to the question I want to answer. And I've presented this to many different classes, and one of the most interesting questions I got was the student asking me, well, how do you know when you're done? And my answer was, well, I've been doing this for 25, 30 years, and so far we've never finished any of those. You get around once, and you think about what the next step is. So you keep refining and getting closer and closer, pushing those two together, but I don't know that it's ever complete. This is just the research cycle and the way I think we want to think about our data analysis. So I also want to go into this pointing out that our perspective as an individual data scientist can impact what we see in the data. Data science can reveal new things, so we may have some really cool visualizations or we may hook together data that haven't been together, but we also have to be open to seeing the patterns or the results that come out. So we can link and process, wrangle, and munge our data, but what does it really mean? So I'm going to give a data science example from 1854. During the cholera outbreak in London. This is before the germ theory of disease was well established, so there were the miasma people who thought it was in the air. You could smell how bad it was in the summer, and that's when it broke out, versus contagion ideas. The miasmist theory at the time was that there was bad air in the storm sewer system, and it was coming out through the drain holes. And then, as many of you may be familiar with John Snow's work, he was thinking that it was the water from the water pumps. So this is big data circa 1854. This is a chart in a book referenced on the right, Cartographies of Disease, of the cholera outbreak in 1854 presented to the London Board of Health. And I'm not sure if you can see my cursor, but this big curved spike on the bottom is the number of cases of cholera along with diarrheal disease. Those two follow each other carefully. Everything in the background are meteorological things like barometric pressure and rainfall and temperature and humidity. And they were doing on this chart a lot of what we try to do on the computer now is try to line up what's the magic signal that starts off the outbreak. So, you know, some of the things we're doing, even though it feels very modern, this sort of scientific inquisitiveness when you're presented with lots of data from lots of sources, has been around for a while. Of course, the big news in 1854 was Dr. John Snow, an anesthesiologist who mapped the cholera deaths in a neighborhood. And I apologize in advance, this slide has sound that was attached to it by Carol Gotway, my co-author on our book, and I haven't been able to figure out how to get it out. But the way the story generally goes is Snow maps the cases in this particular neighborhood in London. He looks at the map, he sees a concentration of the deaths around a particular water pump on Broad Street and says, Eureka, that must be the cause. Snow led an effort with others, with the London Board of Health, to remove the handle of that particular water pump. So the short version of the story, this is Snow did phenomenal work, and reading some of his papers is highly recommended. But this has been kind of compressed into sort of a fairy tale. So this is a travel log for London saying in 1854 Londoners were dropping like flies from cholera until Dr. Snow figured out bacteria were in the water. He turned the pump off, saved countless lives, and now there's the John Snow pub nearby. So that's good information, but it does compress a lot of work that was going on and a lot of controversy. So we look at this visualization of the map. John Snow's map's often presented as, good grief, if you just map the data, look at what you can find. And so it looks like a real triumph of 1854 data science and visualization, but if you look carefully, the maps of the miasmas looked exactly like Dr. Snow's, and if you think about where the drain holes are, they're right by the water pumps, because if your pump leaks, you'd like to have it drain out. So the maps look the same, and in fact, one of the leaders at the London Board of Health had the following response to Snow's 1854 analysis, and he says, this certainly looks more like the effect of an atmospheric cause, or miasma, than any other. If it were in the water, why wouldn't the cholera have prevailed equally everywhere where people were drinking the water? So we've got two different groups of people looking at the same data, the same visualizations, and they come to a different conclusion. Now, history supports Snow's conclusion, and we see that as a triumph, but it wasn't as if he slammed the map down on the table and everybody saw the truth. So that's a nice story. What does this mean for us? There's been a lot of data science breakthroughs in managing, linking, processing, and visualizing distributed data, but you could think about what would someone like John Snow, an 1850s data scientist, do with modern computing? Like if he had a geographic information system or Google or social media, well, you might be thinking he would do something like Google flu trends. Google flu trends prediction goal was to try to beat the CDC's projection of number of influenza-like illnesses over a week, which are reported weekly, and they were doing it for a while. They were about 2 weeks ahead of the CDC release numbers. These are no longer published on a regular basis, although you can still get them. So what happened is in February of 2013, the Google Flu Trends was tweaking its algorithm, or actually a month before that, and started to over-predict. And in a particular week in February, they predicted twice as many cases as actually showed up. So there's a very nice summary of this. It's a short article in Science by Laser et al. Parable of Google Flu, and I spelled flu incorrectly, or it was auto-corrected. So that's, I guess, another bit of big data for you. The way Google Flu Trends works is it's based on correlated searches, and so you're looking for correlations between the kind of searches that happen that correspond to the beginning or intensification of flu outbreaks. And things like high school basketball. Searches for high school basketball scores will start at the beginning of the winter, about the same time the flu starts. So it's not a causal connection, but it does have strong predictive power, and this has been one of the big successes of Google and other data science development areas. Now, the Flu Trends algorithm was proprietary and kind of consistently tweaked. And so it's hard to say that it was wrong all along. That's not true. It did very well. And I'll show you one of the plots from the laser paper on the next slide. So, and the authors don't discount the impact of big data. What they do discount is that, or point out that it offers kind of an example of big data hubris, or we kind of ignore everything we know about flu and just go with the data. So this avoids the subject matter expertise part of the Venn diagram and starts to wander maybe not in the part labeled danger zone, but it starts to get away from as good a job as we could do. So this example to me is kind of an example against the idea of totally hypothesis-free science, and I'm sure those people will debate me on that. And I think we need to worry about hypotheses and past knowledges that we know. I don't know that we have to identify a testable hypothesis for everything we do, but we certainly need to be documenting what we're after and how we plan to find it. This is the figure showing the estimates, and what you see in the top plot is the percent of influenza-like illnesses, and the bottom is just the error between the two, showing the noise around 0. And what you see is starting after July 2011, it starts to deviate some more and into 13. So with things like that, if we find a mistake, it's not so much pointing out a mistake and saying that it's wrong, it's what could we do better with this. So there's several quotes about learning from mistakes. You know, anyone who's never made a mistake has never learned anything new. Each mistake should teach us something, and one of my recent favorites is always make new mistakes. You should learn from the mistakes you make, but don't be afraid to try new things. And so how do we expand data science without reinventing the wheel regarding data challenges? Some of that is knowledge and summaries of these. So in order to avoid mistakes in data science, as we expand, we should carry an appreciation for database stumbling blocks that have been established in the past and explore how these manifest at scale, and keep an eye out for new sorts of challenges that may not have been an issue in older sized data sets but would be now. So I'm going to review 4 data analysis issues that remain relevant for big data but also need to be customized and refined in the big data context. There's a couple of important contrasts I want to mention too. The first is observational versus experimental data. And this is where you're taking data that you are seeing versus data that comes from a designed experiment. There was a period of time where the literature referred to this as found data. Working in geographic information systems, we do this a lot. We'll take the census data. I don't want to map the census regions because they've already done that. I don't need to reestablish the census data, but I want to link it to new data. And message I tend to give my students is the data always looks greener if you didn't collect them. I think we think that about social media data. If you think about electronic medical records, they include a lot of information, but some of it may be opinion, some of it may not be as good as it sounds. So as we grab observational data to make decisions, one question is, does design matter? Should we be hoping for an experiment or link this to it? And does it even matter? I think I gave a big data talk once where my title was Design is Dead, Long Live Design. And my argument was we needed to come up with new types of design to understand the sorts of studies we were doing. That doesn't mean not to take use of the big data, but it's to try to figure out how to make the best use of big data. So some questions, do data have to come from a design to be useful? I don't think that's necessarily the case, but I think you can gain things from a design. And does data quality matter if I can get a lot of data? This is another big argument. If I'd like independent observations, that'd be great, but I could take a whole lot of correlated ones and maybe get the same information. So what do you lose in observational data? At the smaller scale, this is something epidemiologists have been concerned with for a long time. One design data about, I thought this was a really nice example that Xiaoli Meng from Harvard has put together. He proposes an interesting kind of thought experiment. Now this is in a discussion, that the reference is on the bottom, of a paper by Keating and Lewis on the perils and pitfalls of self-selected entry into epidemiologic studies and surveys. The idea of using like web forms, so web recruitment to define your control base and so on. And it raises a lot of question, and there are many pages of discussion after. So if that's an area you're interested in, I think there's a lot of good information in the article. and the discussion. But Jolie points out this example. Suppose we're interested in estimating, say, the average income in the United States, and we had two choices of data sets. One was 100 individuals selected in a simple random sample, so every individual in the United States is equally likely to be selected and would participate. Or we could get data from 50 million individuals, but it's not as clean data that there's a correlation between their income, which we're interested in, and the probability of being selected. It's not big correlation. He was setting an example of 0.05. So what we're saying is, suppose income was slightly correlated with the probability of completing a web survey about your income, and then you want to get the estimate. Well, 50 million and 100 seems like, wow, that's so big, that must be good enough. And Ming does some algebra I'm not going to show you, but it's in the discussion, that there's actually more information in the small sample. You'd need about 62 million to get the same amount of information. So in a big data setting, we do get a lot more, but I think there's a lot of interesting calculations like this one to be done to talk about the value and quantity of information contained in data like that. Also in design versus experiment, design experiments allow one to control outside variables, things like temperature or genetic strain or precipitation, that could have an impact on your outcome, but they're not your primary interest. And in studying flu, say, if we only took observations in the summer, we would miss things like the peak that generally happens in the winter, associations with travel periods and the times when schools start. Design can help remove things, but we also don't want to design our way around important contributors. So we have observational versus experimental data. We'll be talking mostly about observational. The second contrast is prediction versus estimation. I'm not going to make a contrast in my conclusions, but I do want to point out these are different issues, and we'll bring up both. A lot of this discussion hinges on a paper in 2001 on the two cultures in statistical modeling that I think is one of the things that feeds into data science. And the basic idea is if we have input, these might be predictors, they go into a box, something happens in that box, and we observe some output. The two cultures differ in what we're interested in. The prediction group is, give me a new X, can I predict Y really well? And I don't really care what happens in the box. I just want to be able to predict the outcome well. This information side or estimation side is, can I define the associations between X and Y and try to figure out what's happening inside the box? That may be just measures of associations like regressions or logistic regressions, or it may well be something like, more advanced machine learning, deep learning, or a causal inference. So the distinction between the two is what do you care to find out? In epidemiology, there's a little bit of both. We do want to predict how many flu cases there are because, one, it can tell us how to set up a prevention scheme. If we know the types of strains, it can help us plan for vaccinations. But there's a medical interest in what causes things to spread, and we're interested in what happens inside the box too, because if we predict well, I'd like to know what's in the box so I could change the outcome. So what can get in the way of doing either prediction or estimation? How do we get the most out of big data? Well, with respect to prediction, again, the Google flu trends is like we didn't really care that how basketball scores were associated with flu, but we found out they helped us predict better, so that's a good thing. With estimation, what can get in the way is what makes our estimates wrong, and then if they're biased, is bias always a bad thing? So I'll come back to that in a minute. These are the four issues we're going to be talking about in this context of big data and data science. Multiple testing, bias, confounding, and missing and mismeasured data. So multiple testing and statistical hypothesis testing has been a mainstay for small data analysis for decades. Now there's been a lot written about it. It certainly there are some legitimate concerns about turning all of your results down into a yes or no answer, but it's still out there. The p-value reports is certainly something to discuss, but you will see a lot of publications based on that. So in a statistical hypothesis testing setting, you reject the null hypothesis at some level of statistical significance, and typically this is 0.05. And there's probably a lot of talks about why 5%'s there. So this means for each test, there's a 5% chance of rejecting the null hypothesis when the null hypothesis was actually true. So it's a false positive result. Well, it's 5% for each test, but if you're doing a lot of tests, what does this mean? So when we're testing a lot of hypotheses, and these might be checking associations between several outcomes or several variables, you might think of a gene expression microarray, lots and lots of observations. If the tests were independent of one another, then you would expect 1 in 20 to yield a false positive result with this 0.05 level. But what if you're testing 200,000 genes? The magnitude of the problem, there's a lot of highly technical statistical calculations you can do on this to get exactly what it is, but especially if you've got correlation and not independence. But the problem remains, how do you scale this up? So what do you do? Some potential solutions, and some of which have been around for a long time. There's Bonferroni adjustment. If you have k independent tests, you just adjust the significance level of each of the tests by dividing by k, so the overall false positive probability of finding any false positives is 5%. So it's a very conservative adjustment, and it doesn't really scale well. So if I want to split up my 0.05 across 200,000 tests, I'm talking about a p-value for significance of 0.0000025, and that's not particularly meaningful. You think about how much data you would have to collect to have that kind of precision out in the tails to be sure that that's what your probability was. Putting these things together usually gives a family-wise probability of any false discoveries. The probability that the number of false discoveries is bigger than 0, and that's served pretty well. There's a lot of literature on multiple comparisons. My own father wrote his dissertation on multiple comparison issues, and there's a SAS procedure with a Waller option using his technique. But their scalability is a real problem here. In 1995 and refined in 2000, Benjamini and Hochberg introduced a concept called the false discovery rate, rather than the probability of false discoveries. Now, we often think of rates and probabilities as similar, but there is a key distinction here that they were making. The false discovery rate's not the probability of any false discovery, it's the expected fraction of discoveries you make for any cutoff that are false. So this is different than what the Bonferroni family-wise is doing, which is the probability of the number of having any false discoveries being bigger than 0 is 0.05. So the Benjamini and Hochberg is saying, we think it would be worse to have 4 false discoveries out of 10 than having 20 out of 100, where it would be 1/5. So it's the rate of, out of how many you detect, what fraction do you think are false? And this is fundamentally kind of a different way of thinking about it, but one that scales much, much better. And so there's been widespread use of the false discovery rate, especially in the genetics and genomics literature. And there's a lot of resources for exactly how to calculate the false discovery rate. I'm not going to talk about those here today, so we can talk about the other topics. But it's an excellent illustration of how an inferential idea, things like false discoveries and type 1 error rates, needed to be rethought to scale to the big data setting. So we had an adequate solution at a small scale, but it didn't extend. So addressing the issue required backing up a little bit to that in the whirling vortex. What question do we really want to answer here? And kind of starting from those ideas again. There's a really nice FDR overview and some slides that Chris Genovese has posted that I used to help summarize some of the results above. So moving on to bias from multiple testing, the quintessential example of anyone teaching bias and variability, I know this is the case in public health and probably a lot of areas, is this sort of 4 targets here, and you're shooting arrows at the target. Upper left, we've got low bias and low variability, where we average out there in the bullseye, and they're not very far apart. Moving to the right, we're still averaging out near the bullseye, but there's a lot more spread, and a single shot may be further away. Bottom left, we've got high bias and low variability. We keep hitting the same spot, but we're off consistently by some amount. And then the last one, we're not close to the bullseye and we're not very consistent. So high bias, high variability. So this connection with bias and variability is an important one I'd like to keep talking about. But first, this is the New Yorker cartoon version of that. There's something called the Texas sharpshooter approach, where you shoot the side of the barn and paint the bullseye around it. This is the medieval version, where you've got someone to do the painting for you. So some bias we live with every day. So those of you who are parents probably have used an ear thermometer, or those of you who just don't like having a thermometer on your tongue may have used an ear thermometer. They don't give you the same number. They both better measure body temperature, and they're both pretty reliable. They have smaller variability, and you get the similar answers as long as you use it right, but they're not centered in the same place. In this case, there's low variability. You've got reproducible bias, but we can correct for the bias with a simple adjustment. In the case of the thermometers, we kind of memorize what normal is on either one. And then if you're taking your child's temperature, you're also trying to remember, do they seem well enough to go to school or not? Bias and variability come together, and there's this bias variability trade-off in a lot of statistics books. And one of these is if you're talking about the mean squared error of prediction, how well are you doing? One summary of that is taking the expected value of the difference between your prediction and the truth, what you're predicting, squaring that. That's called the mean squared error. And doing some algebra, you find this is the sum of two things. It's the variance of your prediction plus the square of the bias of your prediction, how far you're off. So if bias goes down, variance can go up. For the mean squared error, it can still stay the same. So the accuracy of your prediction is a function of both the variability of your prediction and the squared bias of your prediction. This is an old statistical idea, and it carries in a lot of settings. And sometimes you can reduce the variance by incorporating a little bias. So Tom Lewis has a nice essay on bias isn't necessarily a bad thing, as long as you have some control over it, if it helps rein things in. This is an element of a lot of Bayesian methods, which induce some bias, but they actually add precision. And they borrow information across data sets. I think last week, Michael Newton's BD2K seminar on Bayesian statistics gave some overview of this. But if you look up borrowing information, you'll see how biased estimate can actually give you some improvement over an unbiased one. Let's think of an example of linear regression. A lot of times when you are taught linear regression, you're told that ordinary least squares estimates are unbiased. And in ordinary least squares, you're trying to pick some parameters beta to minimize the difference between the regression model, the x beta, minus the observations y squared. And it's unbiased, but there's an important caveat to that. It's only unbiased if we have all the right covariates in the model. And I put this simple regression picture up here that I made up because when I took a regression class, they told us how to build a least square machine. I thought this was a cool idea. It would be hard to scale this up though, but if I had a board of wood and I nailed in nails where all my points were, and I had a stick and I had rubber bands from each point, and I have to make sure the rubber bands are going up and down with the y direction, but you snap it, the stick, the equilibrium point where all the pressure is on, the stick will land on the least squares regression line. So let's think about that. All of those points are pulling on it with the rubber band, and so it's going to end up in the middle, and it's trying to have the minimum total tension in the rubber bands. But suppose we really needed x1 and x2. So rather than just a single line, there's this plane, and it's the way I've tried to draw this is there's a negative slope going down in the x2 direction, and negative slope in the y direction, but it kind of slopes. You can see we've got clouds of points around it. So suppose we really needed x1 and x2 to do this, but if we only observe x1, I'm going to back up one slide. It's like we're taking a flashlight, shining it down on the x1, y plane. All those points are going to put their shadows there. These gray lines I have, the solid gray lines are kind of the lines in my plane just to show the different groups, the different parts of x1 and x2, but that you really wouldn't see that. And if you fit a regression line, let's say it's this dashed line here. It may be unbiased, but there's an awful lot of noise around it as opposed to the amount of noise that would have been around the plane. So it might be unbiased, but noisier. But suppose we didn't get to see all of the x2s and there was some change in whether they were included or not. So I'm only including the ones in the circle now, which means for the low x1s, I only saw low x1s for the x2s. So the non-circle ones get left out. And if you notice the bar for x2 out here is the higher values of x2. I pick the low ones, and for the higher values of x2, I pick the high x1s. Now I would get a line that goes in the completely other direction. So there's a sampling bit. If there are changes in the chance of things being included, it can bias our results, and it can do it quite a bit. So what happened? It turned out x2 contained important information we needed to consider. In this case, it had information about what values of x1 were included in the data set we got to see, and the result is a biased estimate of the true association between x1 and y. So this is one type of bias that can result in observational studies. If we don't control for the value of x2, but it had an influence on what we were seeing, then our resulting estimates would be changed. Now this is a sort of an example of confounding. Confounding is a big word in epidemiology. I know confounding gets brought up in big data analysis often. Rothman and Greenland describe it as confusion of effects. In the example above, x2 was confounding the effect of x1 because x2 was controlling which values of x1 we saw. Now, confounding, and I've written encyclopedia entries on confounding, and every time I do, I get a headache because it's not easy to define mathematically. There's no single mathematical definition or statistical definition, so it's hard to develop some sort of test or solution, but it is a conceptual meaning we all understand. It's an effect that would confound the one we're interested in. So if we were looking at associations with smoking and lung cancer, age might be a confounder. There may be a birth cohort confounding in terms of level of smoking. So the concept's familiar. There's always room for sensitivity analysis with confounding. You can try to include potential confounders and see whether your results change when you add them or take them out of your analysis. And I think this is a particularly important contribution to the subject matter part of the Venn diagram for data science, because there are often established sets of potential confounders for particular outcomes. In the epidemiology literature, if you don't control for age and race, the referees are going to ask you why you didn't do it. So you could even check and not have it make a difference and leave it out. You will still be asked about it because so many health effects vary with age, so many health effects or health disparities are observed in different race groups, and smoking has always had a big influence too. So I'd encourage you to think about it, but like I said, I've been thinking about it for 30 years and it still is hard to get a handle on. Now I need to click, and there we go. Another example of expression array, and this came up in Rafa Irizarry's BD2K webinar, which is archived. He was looking at gene expression arrays and talking about batch effects. When gene expression arrays first came out, it was an amazing thing. You could measure, responsiveness in multiple genes at once. And so his experiment here he's discussing, or the example he's talking about, is suppose you're comparing gene expression array results and you have some of your gene expressions are from tumor cells and some of the genes are from non-tumor cells, but you send it to the lab in 2 batches, and let's say they ran all the tumor samples on Friday, then everyone left for the weekend, they came back and ran the non-tumor cells on Monday. And if you see a difference in the expression between the different genes you're investigating, what does it mean? Well, we'd like to think it means a tumor-non-tumor difference, but it may well be a day of processing difference. It may take a while to get the lab set up again on Monday, or we may not have things turned on and warmed up, or this is sort of a design issue behind even observational studies. So one of the things you would see in the evolution of publication about microarray analysis or gene expression analysis was an introduction of adding replicates and starting to look for some of these confounders. So if you're interested in more of that example, Rafa Irizarry's BD2K seminar does a nice job of walking through that and showing a lot of examples. Last area I want to talk about is missing data. So what if you're missing outcome values for some predictors, or what if you're missing predictor values for some outcomes? Should you ignore it? I mean, this becomes a frequent, if not ubiquitous, issue in connecting large data sets, is that you won't have all the pieces. Should you ignore it? Should you plug in a reasonable number? Should you just put in the mean and hope for the best? These are all things that are done sometimes, and I think we've all thought about. If you're missing the outcome, one algorithm that's seen wide use on the smaller scale, and we have to see how it scales up, is something called the EM algorithm, the expectation maximization algorithm. So suppose you're missing a few of your outcomes variables, but you have the x's, you have the predictor values in your regression model. So I'll show some graphs of what this looks like. The algorithm, or these algorithms, iterate between filling in the missing information based on your current analysis, your currently fitted model, and then refitting the model, and then updating your estimates and so on until this converges. So in a regression setting, we might have the following. Let's say we have the observed dots, but the 2 I've circled, we didn't really get to see the y values. So I know what the x values are. Those are the dashed lines there, but I don't know what the y value is. Now, if I knew the right answer, I should get the gray dashed line. But if I leave these 2 out, I get a bias. I get the orange graphed line. So the E step is on the left, and the M step is on the right. The E step, you would replace you'd assign the missing values, the expectation from the orange line. This is what we think these two y values should be if the model was right. Then the M step is to put those values in and recalculate the orange line so it moves over. In this case, it got closer to the gray line. And then you'd kind of iterate back and forth with these. You'd be updating what you think y is and updating what your estimate is. And under pretty broad conditions, this will converge to the right answer. So again, just the E step is expected values. The M step is refit the model with updated guesses for missing data, and you repeat till convergence. Now, I'm sure this has been scaled to big data and distributed computing, but trying to take the algorithm and break it down in a way that could go off of distributed processors and so on, it has a lot of validity and a lot of interest. Now, if the covariates are missing, what do? Do we ignore them? We've already seen that ignoring x2 in our example earlier led to confounding and bias because it was important. Could we impute a value or make a guess? What if we could guess a lot of values? Would be value in that? Well, multiple imputation is an area that's been used a lot in smaller data analysis, and has some similarities to cross-validation that gets used a lot in big data analysis. It's just they're not doing exactly the same thing. What multiple imputation does is for every missing value, you simulate multiple values, and then even doing 2 or 3 for each one rather than just one allows you to estimate the missing value itself and the variability in that missing value. So you're not guessing it perfectly, but you're going to have to guess it with some variation and then percolate that variation through to your model. So the imputed values inform us on the overall variability of our estimate or prediction, and they take into account the extra variability we've added by putting our imputation in. So this requires a model for imputation. How would you impute it? How often would you impute? And the Little and Rubin book is really the standard for both the EM algorithm and multiple imputation. I also like Schaefer's book, even though this is it's been out quite a while. There may be a second edition to this. And seeing how some of these ideas could be played out in the big data setting, I think, would be incredibly valuable. So just to wrap up, and then we'll leave us some time for questions if any have come in. Multiple testing, the main issue is if we check a lot of things, we need to control for the impact of false positives. So what do we do about this? We should always worry about multiple testing. I mean, I don't say you should not do it, but you should be somewhat concerned and maybe feel a little bad about it. But you know, the ideas like false discovery rate and similar summaries are gaining a lot of ground. We've got a better understanding of them, and it involved backing up to the basic ideas and building something that was more extendable. And bias is when the issue is the estimates differ from the true values, and what we should do, well, of course, we should worry about that. We should document and adjust if possible, as my ear and mouth thermometer example, but also trying to get, you know, in a more general sense, trying to model the observation process, try to think about how we see the data we see, how that can have an influence on this. So I really think that subject matter component is important in data science. It's not just a calculation and then hope the numbers are right. So I think there's a lot of work to do on thinking about bias, and again, tied into the Bayesian estimation, it provides a way to borrow information that may be a little biased, but it may improve our precision. With confounding, we've got a factor outside of our analysis impacting our results. Again, what we should do is worry. And epidemiology articles are particularly good at thinking about ways that the observational conclusions may be wrong, and how to prove it better. I think sensitivity analyses are always a good idea in terms of checking the impact of things. Now, on a large big data setting, we might not have the flexibility for, say, simulations that we do on a smaller scale, but we might want to think about ways to look for evidence. You know, what wake does confounding leave in your data as it moves through? It's also important to catalog the common confounders in a certain area of application. We shouldn't be doing analyses de novo in every setting, and we should learn from the past. I think this is one of the points that the Laser et al. Article was making about the flu trends. It's fine to make mistakes, but we also don't need to do our analysis completely oblivious of what people know about this area or about the data types. Finally, with missing data, the issue is the data are not complete. This is not a surprise to anyone, but again, we should worry about it, not just brush it under the rug. especially documenting the reason it's missing. I gave you an example with my confounder where I had missing data on x2 that impacted the estimate of x1, and that's because the probability of being included was related to the outcome I was looking at. You have a lot less bias if that missingness mechanism is random. If you have data missing completely at random or a little more generally missing at random, it won't impact your results as much. There are things like the EM algorithm imputation, but I do think there's a lot of work there to think about scaling those up into more general tools and having them fit into the data scientist toolbox. And model missingness, I think there's a lot of room for explaining why the data are missing. That may actually be the first round of analysis you do. If in my example, there was an indication that x2 had an impact on whether the value of x2 had an impact on whether x1 was observed. And if you can understand and model the missingness, this might help untangle the effects it has. So to bring this all together, data and analysis issues that come from statistics and epidemiology and biostatistics, like the four we talked about today, all have deep backgrounds at standard size analysis, but they may not scale directly to the big data setting, and I think this is a very interesting area for collaborations between the large-scale data scientist interests and people trained in statistics and epidemiology. So these aren't issues that one can avoid, so that I wasn't being totally facetious about saying worry about everything. I think worry is essential to doing good work. The Bonferroni and false discovery rate example illustrates how addressing data issues in a big data setting can require rethinking even basic assumptions. It didn't say the assumptions were wrong, it just said the relevant proportion we were interested in was different than what the Bonferroni had been adjusting. So there's a lot of background within each of the 3 or 7 or however many elements of the Venn diagram, but I think the most interesting research advances occur in the overlap, and that's where I think data science will kind of come into its own. So I really like the idea of team data science and having different skill sets available, and as a great framework for addressing this. I think it's more interesting to have a statistician and a computer scientist in the room both thinking about the same problem rather than having them work separately and then present their solutions to each other. So I would say let's go forward and make new mistakes together, and I believe we have some time for questions.
Fantastic lecture, Lance. Thank you so much. We do want to encourage people to write in their questions, and as people are putting fingers to keys, I had a couple of questions, and I thought these are sort of big questions, and I wanted to just kind of get your thoughts on these. You had mentioned one thing in some of your introductory remarks, and a couple of remarks you had made, and one of them involved an interesting term, which I thought was I don't know, kind of caught my attention. That was of data hubris, big data hubris, and you can kind of understand that like lots of people think, oh, if we just have more data, the answers will be in there, and we've got all these methods and techniques to go and just ferret them out, and we'll just know everything. And the hubris element of that I find to be somewhat fascinating. And I think one of the more recent examples of this that everybody's kind of aware of, and I don't mean to get political here, but was in the recent U.S. Kind of elections, where it was clear that one candidate everyone was predicting that candidate was going to walk away with the election. And then on the given night, everyone was a little surprised at how that turned out. And I'm curious if you had any thoughts about, whether or not there was any bias or variance in the processes that led up to that somewhat surprising outcome.
Well, I'd say the kind of ubiquity and ease of doing a survey that may not be a scientific sample survey. I think the polls and so on really took a hit. I don't know if other people are like me, but I'm getting multiple calls at dinner time now that are, recorded surveys on how's the government doing. And in my heart as a statistician, I want to supply data, but in my head, I also realize these are all paid for by different groups, and the wording of the questions are looking one way or the other. And so I think there was a lot of, with the diversified news media and everything, they know what will play to their audience, and so questions were asked in a certain way. I don't think there was as much pooling of the information across the survey. I mean, there's a very complicated setting. There's a lot written about like Facebook and the kind of divergent, you have your group of friends and you forward articles that tell you kind of the same thing. We're all sort of in our own bubbles. I think 538 tries to do a good job of that, but you've got all this information. I think 538 worked really well the last couple of elections because it was based on polls that had measures of uncertainty, and that's what it's good at doing, putting them together. But if the questions are leading and your probability, my probability of participating is not equal for every poll. And I am frustrated by that, but I also, I'm reacting just like a lot of other people are, that I may only be answering polls that kind of cater to what I hope. And I think in that setting, then a poll doesn't work anymore. So that there is kind of this idea that having a lot of data, but finding that it's the people who responded, and that's why the modeling the missingness is really important, and understanding where it comes from. So going back to that data analysis whirling vortex, you know, knowing what data you would like to get and how that differs from the data that you can get or you ultimately get, and what question you did answer with your data. I think we assumed we were answering the question about who's going to win the election, but we were actually answering questions about who's ahead in the minds of those who respond to this particular. You know, once we start putting all the caveats in there, we start realizing we answered a much more specific question than the general one we were interested in.
So one of our listeners writes in, and they're asking about the expectation maximization methodology and whether or not the iterations that you use to fill in the missing data, how often does that lead to a wrong model? Does that make sense? And so why not just ignore those points, you go through this big thing, and it converges on something, and you think, oh, eureka, there's my model, but in fact, it's in fact wrong. How often does that happen?
So it has a good... it has a pretty general set of regularity conditions, the conditions under which it does what it's supposed to do and converges to the right answer. It won't converge to as precise an answer than if you had all the data, because there's some uncertainty in those missing pieces, but it should move in the right direction. There's a fair amount of literature on the, just for lack of a better term, calling the small data setting of its performance and how well it works. You know, I think one of the biggest questions for some of these statistical techniques and rolling them out to the big data setting is, could you distribute it and put it back together? So there's it's sort of like MapReduce, except it's doing the analysis on that. So that's there's used to be, had a different name, but Bill Chambers is doing a lot of this. Of what kind of statistical algorithms can be broken into pieces that they can be sent off to different processors and then recombined in such a way that you keep the answer. So I think that's a very active area for work. It's wide open for good ideas, and I think, you know, that's an area where pairing a statistical researcher, a methodologist, with a computer scientist could lead to some big improvements in both areas, and that the joint improvement of both areas is where data science gets better.
Great. Another question I had was, I see amongst a lot of younger investigators, some of the postdoctoral fellows that I've encountered, there seems to be a particular, value put on finding ever more smaller p-values. And so, you know, somebody will go find something that's, 1 times 10 to the negative 16th and get very, very excited because, well, it's much better than, 1 times 10 to the negative 4th. And so they think it's more important, and they make the classic mistake, I think, of mistaking statistical significance with practical and theoretical significance. They may only be accounting for like 3% of the variance, but it's wildly significant because they've got big data. What sort of message can we kind of convey to people to maybe avoid some of the issues that come up when you fall into that sort of trap, if that makes sense?
Yeah, I mean, I think the easy trap to fall in with p-values is the smaller one must be more significant. And, one of the things, there's been some pushback. Some journals won't accept p-values anymore. And I think part of that is because I think p-values serve a purpose by themselves, but I think they're overused and overinterpreted, and this is one example in which that happens. Even if the null hypothesis is true, the actual p-value you get should be uniformly distributed between 0 and 1. So, ranking them, if you're not rejecting, doesn't make sense, and then the distribution shifts. I'm saying a lot of words. What I think would be helpful is if we start thinking about, measures of association, and then bringing in the subject matter aspect of the Venn diagram. Is the difference we saw really meaningful? Because there can be statistical significance, but the difference you actually detected was kind of small. I don't like to see these p-value contests, like you said. I think that's putting too much emphasis on something that's hard to estimate. And to me, a p-value of 10 to the minus 6, I don't even know what that means, because I've never really observed too many things with 1 million observations to get this, 1 in a million. And if you get these really tiny ones, I think weird things happen, and that we don't appreciate that even though it had a small probability, it still could happen.
Yeah, I agree. I find that, when people, at least in certain areas of big data research, start talking about p-values that are, 10 to the minus 16th or something. I mean, they're down in the machine precision of,
right. Yeah,
and the errors. You just can't trust anything that gets that low. And the emphasis being on the p-value, I think, is somewhat misguided. And I'm not quite sure how that happened, because I was always kind of brought up to think, how much of the variance am I accounting for example,
right.
To give me a sense of how strong my model is. And that ends up not being very important to some people. And, maybe I missed a memo, but it seems that that's where people's focus.
I think it's an example of, data science is a new field with people coming from other fields. And that's why I was talking about the team data science, because this concept that you carry with you, or sort of what you train to look for. What was important about doing the analysis? And having people with different perspectives, I think, is a broader image of that. And so, there's people who think all of statistics is in what they get in one class. Similarly, there's people who think all of computer science is what they got in one class. And we really need to recognize that's not the case. And it's very hard to be an expert at both of them. But you can learn a lot of things from experts in them if they're getting together and working together. So I think data science is a highly collaborative field. I like to think of statistics as highly collaborative, but data science, I think, is often even more so. And it's a combination of people with different intuitions, expertise, experience, kind of default assumptions. And that's where I think we can sort a lot of things out with these kind of collaborations and conversations.
I'm really glad to hear you say that, because my kind of last question I had was, how can we help universities and university administrators kind of understand that team data science is important? It's not just statistics, it's not just computer science that most universities already have, but it's something that's unique and special. How can we help them to recognize that?
I think it's good to highlight the successes in places that have a lot of universities develop institutes of data science, for instance. Some of those institutes are within computer science, or they're within another field, but I think the really strongest ones, and the ones that I think are making the biggest difference, but will ultimately, and I hope this is the case, continue to make a big difference, will be the ones that have this interdisciplinary feel. And I have gone to a lot of statistics conferences where people say, well, aren't we data scientists? That was the early conversation. The conversation I heard last week at a statistics conference was, we're part of an institute and we have a track on statistical methodology, or a track on health analytics. Because we work in a lot of health area, we can help put the algorithms in perspective and what kind of questions they can answer. And we sort of establish some, not standards, but usual ways of doing things. To me, I think that's a big development, because that would argue against having a center for data science inside a statistics department, or a center for data science inside a computer science department, without talking to each other. So, I think as we see developments happen that have benefited from the interdisciplinary collaboration, I think that's where we'll really, I kind of hate to wait for that to happen, but I think that's where the big developments will be. And so as those happen, and also, I am a little afraid that some of the purely algorithmic approaches are going to make some big mistakes somewhere. And maybe it's not just guessing how many people had flu last week. Maybe it's predicting drugs for drug assignment for cancer in precision medicine. If you use a machine learning algorithm that works really well for getting pretty good increase in your profit in a business setting, and you apply that to blindly as a black box, and I don't know if anyone would do that, but suppose you did, to defining that, you may get 90% of the things right, but you've got 10% of patients who are getting the wrong dose, and that could be fatal, it could be costly. So some of this is, I think statisticians are sort of inbred worriers because of variance. And we think about all the kind of errors we can make. We give them names, type 1, type 2 errors. We need to develop kind of what can go wrong, and that's why these 4 topics came up today.
Well, let me thank you on behalf of all of our listeners today for your fantastic talk, Dr. Waller. Thank you very, very much. Once again, we'll be having another one of these next Friday at 9 a.m. Pacific and noon Eastern, and we look forward to seeing you all again then. Thank you again, Dr. Waller.
Thank you very much.